Welcome to the third year presentation update for my PhD research in biogeochemistry in the Cobar region of New South Wales. My name is Joe Schifano and I'm researching at the University of New South Wales under the primary supervision of Professor David Cohen, co-supervisor Dr. Neil Rutherford, and industry supervisor Dr. Chris Folks from the Geological Survey of New South Wales. The subject of this annual presentation update is pining for an anomaly. Now, through Minex, we have quite a few survey participants and major sponsors. In relation to my PhD research, it's the New South Wales government through the New South Wales Geological Survey. Other supporters of my project include the CSIRO, Olympus, with the loan of the portable XRF Phantom model, and of course the University of New South Wales. Now, my PhD is based on biogeochemistry in the Cobar region. I've been working out there for the last 3 or 4 or 5 years, collecting samples across the basin. As we can see on the right-hand side here, Cobar is about 700 odd kilometres northwest of Sydney in Western New South Wales, and my project area is centred around the Cobar district and the Cobar Basin, from Byruck up in the north, Nyngan in the east, down to Fifield and Condobolin to the southeast, across to Mount Hope and up to and west of Cobar. These blue dots here are all sampling locations for the cypress pines along roadsides, which makes it quick and easy to access without worrying about property incursions. The area is around 42,000 square kilometres. From top to bottom, it's about 300 kilometres. And this major cross-section from Nyngan through Cobar and west of Cobar is approximately 200 kilometres wide. Essentially, the size of the project area is equivalent to the size of the country of Switzerland, if we flatten the mountains out. Today we'll be looking at a couple of spots that we're going to talk about. One is the nickel response that we've found at Miandetta, at a former quarry site. And the second location we'll be going to will be the Hera mine site, where we'll look at a couple of tree dissections, which I mentioned at the end of last year's presentation in November, and we'll look at those today. So the aims of the thesis is to establish the regional background and the mineralised signatures in the cypress pines. That way we can basically separate the two and recognise anomalous signatures in the biogeochemistry of the cypress pines when we come across them. Second major aim of the project is to look at how quickly and efficiently we can analyse the pine needles out in the field and get some meaningful results to help us quickly target in on potential mineralised zones and underlying mineralisation while we're out there on the go. And that is a overrepresentation of a Olympus handheld XRF Phantom model. And I will show you exactly how we sample those pines and get some chemistry numbers throughout the talk. That's a typical cypress pine, about 7 to 8 metres tall. Kind of looks like a bit of a Christmas tree. And we just walk around the base there and grab the needles off the tree. The third part of the project is working with the Geological Survey of New South Wales to develop a database for their online portal for exploration data called MinView. So once all this data has been finalised and databased, hopefully over the next 12 to 18 months, maybe even quicker, it'll be all uploaded onto their system for access for everybody. Now, as I mentioned, we're going to look at the Miandetta area in Western New South Wales. Site 1 here, this location is about 20 kilometres west of Nyngan on the Barrier Highway. And what we've discovered there from the analyses of the pine needles is that we've got some extraordinary high nickel values off the cypress pine needles, and also some test work on the open and closed cones that we can see down the bottom here, as well as the twigs. So when we walk around the tree, we sample the needles, but because we got some really, really high nickel values, about 100 times the expected value in cypress pines, I've also just mucked around and sampled a few of the closed cones before they mature and open. We can see the open one cones here in the center photo. They look like they've become perfect dust trap once they open up. They catch and collect a lot of dust. So we'll see if there's how that is reflected in the biogeochemistry. And I also analysed those twigs that are directly adjacent to the where the pine needles sprout off, up to about 5 millimetres. So in terms of what's nickel's role in plants, it's sometimes considered as a micronutrient for some plants. And nickel hasn't if you look in the literature, hasn't been used all that much as an element, a targeting element for biogeochemistry, but that may soon change with what the cypress pines or Calyptra squarcifera are telling us. Now, if we just zoom in here on the site and just have a look what happens sampling-wise. Originally, some work was done along Munda Lane here, the access unsealed dirt road to the former quarry site here at Miandetta. And an honour student, Agustina Lorby, under the supervision of Dr. Ken McQueen, undertook some sampling. These green triangles here were some of the samples that he took along Munda Lane, and he popped in over the former quarry site and took a few samples there. So there was a little bit of nickel response there. I thought, oh, I should go back and come over the top of those samples and reanalyze it and see if the nickel values hold up over time. So I did that in November last year, where these pink soil sampling sites are, for a first pass look, and the nickel results came back pretty good. In line with some of the results at the Sunrise nickel cobalt deposit down near Condobolin and Fifield, but I wasn't able to get access into the site until about March earlier this year, once landholder access was approved. Once I got access, I've come in and put almost a grid across the whole site. We can see that there's a magnetic response here in the underlying image, showing that the whole area has got a slightly higher magnetic response, and that's due to the underlying what we call ultramafic and mafic rocks, which are generally a bit higher in nickel and cobalt than these surrounding rocks in blue here, which is sedimentary rocks. So we sampled these areas in here. We've got a fault that kind of comes up through the middle here. And if we look at the results down at the bottom here, we also did some soil sampling just to check how much is in the soil, how much nickel and cobalt and other elements are in the soil compared to what's being taken up by the trees. And we can see west of the fault in the bottom left down here that the darker contour of orange there shows that we've got some nickel concentrations here up and around the 700 to 800 to 1400 ppm or parts per million. And on the right, on the east of the fault, much lower grades. Now, once we analyse the cypress pine needles off the trees, we can see that those big blue dots there correspond with up to 120 to 180 parts per million nickel, which is extraordinarily high, about 100 times what our normal background values are, for about 1 to 2 parts per million of nickel. And they coincide with that contour of the higher nickel in soil. On the east of the fault, barely any nickel in the pines at all in comparison to what's on the western side. And we see the same pattern here for cobalt. Now, the second part of the thesis aim there was using the field portable XRF in the field. So I'm just going to run you through a field sampling video here for about 7 minutes, and we'll look at some of the data spliced into that video as well. We'll also look at what happened down at the Federation and Dominion deposits with some of with using the field portable XRF in the field. So I'll just run this for you.
Hi everybody, my name is Joe Schifano and I'm undertaking a PhD in biogeochemistry at the University of New South Wales with the support of MINEX CRC. I'm under the supervision of Professor Dave Cohen and Dr. Neil Rutherford. Today we're at a site called Miandetta, about 20 kilometres west of Nyngan in the Cobar Basin. Now, the Cobar Basin is an attractive area for mineral exploration and hosts numerous world-class copper, gold and base metal deposits. Behind me you can see that is a very healthy looking cypress pine, otherwise known as Calyptra squarcifera. And part of my research for the last 5 years has been sampling cypress pines across the Cobar Basin with a view to using the needles that are taken off the trees for analysis for metal content to help us find and vector in towards mineral deposits in the substrate. We'll just go over and have a look at a tree. Let's flip around. So generally, I'll work my way around the tree and grab a bunch of cypress pine needles every so often. So I'll work around the tree, making sure we don't get any of the cones, and grab just total needles. That's all we're looking for. Now, once I've been around the tree and grabbed a whole bunch of needles, I've already collected a bag here. And essentially, it looks like that. So that's about 100 grams of cypress pine needles. And the first step is to use a portable field analyzer, otherwise known as XRF. And what that does is give us some initial indications of what the geochemistry might be. Now, this particular site at Miandetta, we discovered that the cypress pines, based on the underlying geology, where we've got some elevated nickel in the rocks below, are very receptive to the uptake of nickel. So I'll just put the XRF inside, and I've rested on top of the needles there. It's about a 10cm padding of fresh needle just collected. And we're going to run an analysis. That, just turn it on. And away we go. So straight away, we can see nickel is running at about 166, 65 ppm. And that is very high. And we know that holds up well with laboratory analyses, because we've already sent some of these off to the lab and then essentially come back at about 160, 170 ppm. So what this does is help us vector towards mineralization and tells us that the cypress pines are an excellent sampling media for searching for mineral deposits. Now, I've also, for completeness, collected some of the cones, some of the bark, and we'll submit those for analysis as well, just to see where all the nickel is concentrating within the tree. We know from previous work that it's the needles that seem to be the best part of the tree to sample. Now, this spot was great for nickel. Our next stop, we'll look at a couple of deposits south of Cobar, which are going to give us some good copper lead zinc signatures. We'll see you then. So just a quick look at how it all plots up on screen. We've got the Miandetta historical quarry site here. And down in the southeast corner here, we've got a former quarry. The workings are here in this little brown mound, and we've got a fault running through from top to bottom. Now, the nickel samples in the pines left of the fault or to the west are all very high nickel, and the tree that I was sampling was just down in this southwest corner. And all the trees over onto the right-hand side of the fault with those small blue dots are very low in nickel values. So there's a big contrast from both sides of the faults there. The nickel values generally only around 2 to 3 parts per million in cypress pines, and that's from over about 1,300 that we've analysed. Yet here at Miandetta, we get some extraordinary numbers up to 150, 160, 170 ppm. Hi everyone, we've finally reached our final stop at the Federation deposit, and today we're just east of the Dominion prospect because Federation is being drilled out and we've lost our trees there that have been cleared. Now, I've just zapped that tree after collecting my pine needles, and we're going to have a look at the importance of lead. Now, if we just come down and have a look at our field portable, I've just run this sample, and we can see down the bottom there, we've got a reading of lead 6 ppm with the field portable XRF. Now, the importance of lead is that we only really see any lead reading with a field portable when we're in the vicinity of mineralization, and we generally get a bunch of trees that are tightly clustered, giving us a lead response. Once we get away from any zones of mineralization, we go quickly back to background, which we can only get a reading of that through the lab results, which is about 0.4 ppm. So the importance here that we've discovered using the field portable, and specifically with lead, is that we only get a reading of lead or some numbers when we're very close to mineralisation. So that is a great help in vectoring towards mineralisation and deposits under cover. So now that we've sent all our pine needles off to the lab and we've got them properly assayed, we can see here for the lead response over Federation and Dominion that we get some excellent contrast with the background pines, all these other trees that we've sampled. Now, if we can contrast that with our field work, we can see that the XRF was very useful out in the field and gave us some very discrete spots to hone in on in terms of searching for mineral deposits, and a very good contrast with the background. If we compare lead with, for example, copper, we're not getting much of a contrast here. We wouldn't know where to start looking. And if we look at zinc, similarly, same result. So the real benefit here is a powerful element, lead, that allows us to hone right in as an ore finder for these mineral deposits using biogeochemistry. So in conclusion, what we've found so far with the research is that the pine needles could just be the needles in the haystack that we're looking for when exploring for mineral deposits under cover, especially using lead. And there are a few other elements that are working well, especially with our laboratory analysis, including tungsten. So if you've got any further questions or interest, I'd be happy to hear from you. Thank you. Bye.
Okay, I hope you enjoyed that quick little field sampling tutorial. We can see here on the right-hand side, the nickel value was very high at 167 on the field portable XRF. Now we'll just jump in to have a look at those needles and twigs and cones that I mentioned that I sampled. We finally got the analyses back for those. So I'll just jump back a bit. So we're going to look at tree number Miandetta here, and then we'll also look at similar results for tree number 32, which is about 30 metres way to the west. So if we start here with nickel, we've got a massive number here of 177 ppm. The normal average for a nickel response in the cypress pines is between about 1 and 2 parts per million. So extraordinary numbers here we've discovered at the Miandetta site. Now, when I sampled those twigs, even 15 parts per million is still high. However, the big surprise was both the closed and open cones. 108 ppm for the closed cones. I showed you one of the green ones up on one of the first pages. And then once they sprout, dry and open up, even the open cones still showing quite a bit of nickel content. And then even the bark at 67, still big numbers, but the issue with the bark and the problem with the bark is that the bark has a tendency to trap and catch a lot of dust in those crevices, and it's got little furry kind of hairs in there, and they trap and collect a lot of dust. So we generally see lots of extra geochemistry or biogeochemistry in the dust. A lot of windblown dust gets caught in there. Now, if we look at iron, even slightly below average was the iron. We normally get about 300 or so parts per million of iron, so we had about 252, so that's close enough. Very little in the twigs and cones, a lot in the open cones, and maybe it's acting as a dust trap for the windblown dust that comes through quite often. You get lots of wind blowing up through the western New South Wales, and they may be catching some of that in those open cones. As we mentioned, the bark loaded with iron, and we're not sure quite how much of that is actual dust, but there's quite a bit of iron in the bark. And we see elevated chemistry in the bark for a lot of the elements. Next is arsenic. So arsenic generally averages about around about 120 parts per billion in the cypress pine needles. Again, we're a bit below average here at Miandetta. Twigs were pretty low. The closed cones, barely anything. The open cones, there's a big jump when we get to the open cones and also in the bark. Now we know that arsenic can act as an insecticide in plants. So the obvious place for the arsenic to accumulate would be in the bark, which is the first line of defense for any borers or insects trying to get into the wood of the cypress pine. If we come down and look at cobalt, we often get cobalt associated with nickel deposits, or in the ultramafic rocks like we have at Miandetta, we get elevated nickel and elevated cobalt associations. So we can see that our average cobalt down the bottom here is around 230 parts per billion. We've got about 5-6 times that at Miandetta. So a lot more cobalt being taken up compared to what it takes up on average. Still in the twigs and the cones, we have a lot more in the open cones, and once again the bark is loaded with cobalt as well. Now copper, generally copper is fairly well controlled in its uptake by plants, and we can see that there's a pretty flat trend here across the needles, the twigs, the closed cones and the open cones, and always seem to be a bit extra in the bark. So generally, cypress pine needles only want about 3 to 4 parts per million, or about 3,000 to 4,000 ppb of copper, and it seems to be very well regulated. Now, manganese is an interesting one. Manganese does scavenge a lot of other elements in the soils, so possibly we're seeing that it might be scavenging the nickel and assisting in the uptake. So we've got to do a bit more work on that, so that we get above average nickel, manganese in the needles, and then we have an upward trend towards the bark. So we've still got quite a bit of manganese by the time we get out to the bark. We'll have a look at another tree, and we've got that tree number 32. It's about 30 metres to the southwest of number 6, and we have almost identical patterns. For nickel, we almost have an identical amount, about 178 parts per million, which is about 100 times the background, as previously stated. Iron, slightly below average again, but once we move out towards the bark, we can see that the bark's got much more iron in it, and probably that's a function of the dust as well, and also the open cones. Arsenic, very similar to the previous one. If we just look back here at number 6, it's almost an identical pattern and identical amounts. So arsenic stored in the bark, acting as a first line of defense and insecticide. Cobalt, again, not as high as the previous tree at about 1500, but still elevated, 3 to 4 times background. And so it's a similar pattern for also copper as well. We can see we have a flat profile until we get to the bark, highly controlled, around 3 to 4 parts per million. And then when we get to manganese, once again, it's almost an identical pattern as what we saw. So we're seeing a few trees, or a couple at least, through their cones, their bark and their twigs, having very similar patterns associated with a high uptake of nickel and cobalt. Now, the second part of today's talk is to look at a couple of tree dissections. As I said, I mentioned at the end of last year's 2020 presentation that we'd be looking at the results from the tree dissections. We've now got almost our full database of regional tree samples and mine samples, so we can relate what the data for these trees are compared to our complete database of samples. So just to set the scene here at the Hera mine, this is a satellite view of the mine. We've got this surface infrastructure here for the underground mine in the southeast corner, the tailings dam below that. We've got our background tree out here near the mine village in accommodation, flat ground, about 313 metres above sea level, lots of runoff across the ground, a bit more soil cover out here. And when we come back to our high grade tree, which is sitting on a hill, That's sitting about 30, 40 metres higher than the background tree. So we've got two different environments here where we've cut down trees to dissect them. One's much closer to the mine site, and the other one's much further away. In the mine site here, we've got the run of mine pad. That's where all the ore is brought up from underground, stockpiled, and it goes into the crusher and gets processed. We've been out here and done some dust sampling just to get an understanding of what kind of dust contamination there might be. We think the limit of that dust trail is about 400 metres, mostly towards the northeast. We've done a lot more dust sampling further to the north as well. And the outcrop is these pink shades where we've got predominantly more outcrop as opposed to soil cover. This tree here, the high-grade tree, is just on the side of the hill, about halfway up. And we'll now have a look at what we actually analysed. We've cut the trees down, so you can see the high-grade tree here on the left, a lot of rocky outcrop on the hillside. Our second background tree, we're out in the flat area here, about 1.2 kilometres out to the west. We chose this high-grade tree because we knew it carried a very high amount of gold in it, 28 parts per billion, as opposed to our background tree, which is about 1. Still a little bit high-ish in general for a background tree across the whole basin. They're normally about 0.6 parts per billion, but we're in a zone where there's a mine and lots of mineralized rocks surrounding that, so it's a bit above average, which is to be expected. Now, once we dissected or dropped these trees, we took regular cuts along the branches all the way up on one section of the tree. We cut some wedges in. We took the branches, we sampled all along the branches, and we can see the different sizes we took along the branches out here on the right-hand side. We took 4 fractions from where the needles start all the way back to the trunk in various size fractions. And we took slices out of the trunk. So we took the hardwood out of the center. We took the outer wood, which is the lighter colored wood, this dark rind around the outer wood, which is the cambium. And then we also sampled the bark as well. So you can see that in cross section down the bottom. And this one here was from 1.5 meters up the trunk, and you can see what it looks like at 2.1 meters on the right-hand side here. So what we're going to do now is actually look at the results in the geochemistry. We're going to step out of the presentation and we're going to jump into an Excel spreadsheet, and we're going to have an interactive look at the chemistry of these parts of the trees that we've sampled. So if we look at these graphs here, just to start with, on the left-hand side, we've got an XY scatter plot. We've got the height of the tree, which for the the high-grade tree was about 7 to 8 meters, which is typical on rocky outcrops. On the bottom, we've got metal concentration. And on the right-hand side, we have the organ types. So we've got the squares for the heartwood and outer wood, blue and dark blue and light blue for the outer cambium outer bark. The twigs are all circles in order of size from smallest to largest. And we have our needles in green diamonds. On the right-hand side, we've got the same setup with the background tree. Over here, we've got a box and whisker plot, and this will interactively change as we change element. And this will show us the highs and lows on the thin line, and the box in the middle will give us the 25th percentile up to the 75th percentile of the data set of the results. So if we start with gold, our average for gold is about 0.65 parts per billion. Normally in our regional trees, and we get that from about 1,500 regional trees that we've sampled across the basin. So we start with a background tree in the center here. We can see that the green needles, the green diamonds here, which represent the needles, are showing about between 1 and 2 parts per billion. So it's a bit above average for normal, but we are in an area where we're near a mine site and we've got slightly elevated geochemistry, even in the background. However, if we look at the high-grade tree, we can see that on the scale on the bottom here, we've got between about 15 and up to about 42 parts per billion gold. So that's very high, extraordinarily high. So that's why we came back and chopped down this tree to have a good look at where all the rest of the gold is in the other organs. So if we look at what the twigs are doing, all these yellow circles, we can still see we have well above average gold in the twigs, lesser in the heartwood and the outer wood and the cambium. So by far, the cypress needles were the best organ to sample for looking at gold. Even with the background, we still got separation from the twigs, and the needles were still the superior organ to have a look at. Now, let's have a look at nickel, and we can compare that with the Miandetta site that we got the big nickel numbers from. So we'll start with the background tree. We can see that probably the most consistent organ to sample all the way up the tree was still the needles, a bit of scatter with the organs, with some of the twigs and a bit of bark showing a bit of an outlier here. The most consistent one would have been the needles. And that was around what we expect in average, about 1 to 2 sort of parts per million for nickel. Now, if we come back to our high-grade tree, we see a big separation of the needles away from the rest of the organs. Still, the needles were the best organ to sample at about 3.5 to 4.5 parts per million. Still above the regional background, but not as much as Miandetta's. And we can see that maybe the twigs directly adjacent to the needles, before the needles start sprouting out, might be the next best organ for nickel. So we can see in the box and whisker plots there that the high-grade tree versus the low-grade tree, that the needles compared to the rest, probably the best. And we saw that with gold quite clearly. Now, also at Miandetta, we looked at the cobalt. So we'll come in at here and look at what happened with the cobalt here. Cobalt, not so clear cut. So if we look at the background tree, we can see that the cypress pine needles plot in amongst the twigs. So those twigs towards the directly adjacent to where the needles are sprouting out, the lighter coloured ones, they may be more effective as a sampling media when it comes to cobalt. Still, we get a nice straight line with the needles. We've got the bark doing something out here. Possibly a lot of dust contamination from the nickel-rich soil over the nickel-rich rocks, nickel and cobalt-rich rocks. If we look at our high-grade tree, we can see similarly, we've got a lot of the twigs probably just as effective or more as effective as sampling organ compared to the cobalt. So we'll go and look at a couple of other interesting elements. Talked about lead and the importance of lead at Federation and Dominion and other parts of the Cobar Basin where we've looked at deposits. So let's come down and bring lead up. If we start with our background tree, we can see that the needles plot in and amongst all the twigs. The wood and the trunk area, not very effective, highly controlled how much, they don't really want much lead in the wood and the trunk. And the bark, well, the bark does show some trend as we get up the tree, but the needles are scattered throughout all the twigs. So when we've got very low concentrations of lead is not that all effective in the needles. However, when we come across trees that are in zones of mineralization, we can see a clear separation from the rest of the organs. By an order of magnitude, we've got very high lead response here. The Hera mine is a copper, lead, zinc, gold underground deposit, so it's no surprise that we are getting a big kick up of lead here, but this is a very big kick. We're starting at about nearly 19 parts per million out to about 42, and by far very effective. And this shows up when you're using the field portable vanter in the field as well. Once you get a lead response, you start to get very excited and you start closing in your grid and trying to find, tightly clustered trees that might be indicating that you're close to something down below that's of interest. We can see that on the box and whisker plot. You can see that the high background tree is clearly separating itself from the rest of the organs. However, the needles, probably down below where its average is probably about only 0.4 ppm in background trees. It's a little bit above here once again because we are at a mineralised mine site with rocks carrying a bit more geochemistry than normal. Let's have a look at sulfur. Now, sulfur is very interesting. We see separation in both the needles in the high and the background tree. However, we don't see any separation in the values. If we look down the bottom, we're at about 1200 parts per million in a high-grade tree, and we're centered around the 1200 parts per million in a background tree. So essentially what's going on here is the tree saying, it doesn't matter whether we're in a highly mineralized zone or a background zone, we generally only want about 1200 parts per million of sulfur, and that's it. So we're not seeing any increase in relation to the proximity to a mine or being over mineralization or being a tree that's carrying a lot of geochemistry. It's controlling the uptake of sulfur to exactly what it wants and where it wants it. So it wants about 1200 in the needles. And the reason being is that sulfur is essential for all plant species, and it's a component of the proteins and enzymes, and that's why it's managing its uptake to the needs that are required by the plant. Similarly, if we look at another macronutrient, we'll come up and look at magnesium, and I quite like magnesium. We see quite another different signature here for one of the important elements of tree growth. Now, look how tightly controlled in both the background and the high-grade tree that the uptake of magnesium is in everything but the pine needles. It's literally saying that we want 1000 parts per million of magnesium, and that's about it. The rest of it gets sent to the needles, and that's in the order of about 2 to 3, nearly to 4000 ppm. And there's a very good reason for that, because magnesium is essential in the physiological processes involved in chlorophyll. It's an activator for enzymes, and the photosynthesis occurs in the foliage. So that's where they want to send the magnesium to, and we can see that clearly. So although we get a separation as being the best organ to look at, there's no difference because the uptake is almost the same, and we saw that for sulfur. So when we're looking at the essential elements that trees need to function and grow, they seem to be selectively controlling their uptake almost to the parts per million. Now, an interesting side study that we've done to look at that is we've done seasonal sampling. So I've been sampling every season for the last 4 or 5 seasons, so we can see if there's any variation between the seasons. So that's something I might talk about at next year's annual conference once we complete our interpretation of all that data. So we'll see if the magnesium or the sulfur varies throughout the year in the uptake, as well as the other key elements that we're looking at for mineral exploration. So we've looked at a couple of elements that look good, nickel and lead in terms of searching for buried mineralization. We've looked at a couple that are important to plant and tree growth in the case of the cypress pines here at Hera. So let's look at a couple of elements that maybe not so good. We touched on the copper uptake at Miandetta, and it was pretty flat. We can see that almost in the box and whisker plot. We've got almost a flat trend across here. If we look at our plots here and look at the both the background tree and the high-grade tree, we can see a tightly controlled almost bunch of all the organs within about 2 to 3 to 4 parts per million copper. So the needles in the background tree probably sit almost in amongst all the twigs. So there's not that much difference. So probably maybe in those twigs just before the needles sprout are just as effective as using the needles. A lot more fart-assing around to try and collect the twigs, so the needles are just too easy to do. So that's why we keep using the needles. If we look at the high-grade tree, obviously with much more chemistry around in the rocks and soils in the high grade zone, we see a kick up of copper in the needles to probably 4, 5, 6 ppm, and a bit more of a spread in the rest of the organs. So it's still being tightly controlled within a range. So copper doesn't give us that great of a contrast in terms of values in a background compared to the values in a high-grade tree. However, the copper story is not over here. We're still looking at some other sites which potentially might be giving us copper signals. We'll come back to that maybe next year and review copper. We'll look at another element that maybe is a bit tricky to use, and that's zinc. So in a background tree, we can see the average for zinc is around about 16 parts per million. At HERA, we're looking at about between 20 and 25, and it does separate itself from the rest of the organs. So it probably is the best organ. In the mineralized tree, we've got so much zinc here. We're up at about maybe averaging about 60, 3 to 4 times what we normally get, but we do see a kick up in the zinc in those twigs near the cypress pine needles. But it's not that consistent for the twigs. So the needles show a more direct or a more linear trend towards the top of the tree as being more consistent as an organ to sample. So that's been a bit of a look at most of the interesting elements. We'll just look at one more, boron. We'll come up the top here. Look, boron B. Boron's important in plant metabolism. And we do get elevated boron in SEDEX type seafloor deposits. So we can see, again, one of the trace elements here, it's separating itself from the rest of the organs, but the overall values are not different between a high grade and a background tree. So the average is about 30 regionally. So even with a background tree, we're a bit below average here. And with our high grade tree, probably average around the odd 20 there. So the plants do have some control on their element uptake in terms of what they do and don't need. On the elements that they're pretty selective about, like magnesium, sulfur, and we can see boron here as an essential element. They take up a certain amount, and then they probably don't want any more than that. Whereas other elements like nickel, gold, lead, when there's abundance amounts in the ground, they are taking up quite an amount of those elements, which is fantastic for explorers because we can use those elements to vector in towards mineralization. So I hope you enjoyed that. We'll jump back out into the presentation and continue on. Let's get that going. So here is, we've been through all of this, so we'll just jump through the next one. All right, so for those of you that are still pining for more biogeochemistry, in terms of recent publications and presentations, I've contributed a little bit to my supervisor Dave Cohen's paper. He's been working on the pines in Cyprus for the last 10 years and soil sampling there, so I've assisted with some of the XRF working and analysing in that. More recently, we put out a paper just in July in Minerals on the nickel uptake at Miandetta in the cypress pine. So the link's down below there. If you can click on that in this presentation, it'll be available for you, or you can copy and paste or type that in online. Now, if you want a bit more detail on the importance of some of the other elements that we haven't looked at this time round, I've previously given a fairly comprehensive talk to smedge.org.au in October 20 last year. So that's about a 53-minute talk, covers more of the other ones like copper, lead, zinc, at some of the other deposits and discoveries by Aurelia and Peel Mining in the Cobar Basin, at Waggatank to Southern Nights, Malleebull. So there's the Zoom link, or you can go to smedge.org.au, scroll down on the bottom right in the front page, you'll find past events, click on that and you'll find that link on the next page there. Just recently, my supervisor, Dave Cohen, gave about a 20-minute talk to Chile Explorer in September 21, and that summarises some of the research work from my projects, other work from New South Wales with biogeochemistry, and some work from Cyprus if you want to chase that one up. The link is down here. If you can't run that directly from this presentation, there should be a file attached with this presentation that you can jump back out and have a look at later on. So in conclusion, using biogeochemistry, it's a low cost, low impact, rapid sampling method. So depending on how close you're doing your spacing, you can get 60 to 100 samples per day. We've done more in some incidences. It has applications in areas where you've got maybe shallow to deep transported cover and you don't think the soils that are effective. If you've got transported cover, those tree roots can get through that transported cover and tap down into the underlying bedrock and maybe get closer to some more mineralised anomalies and signatures. So cypress pines are showing to be excellent sampling media for vectoring towards mineralised zones, especially for lead, tungsten, cadmium sometimes as a proxy for zinc, antimony and arsenic, using ICP-MS, and also we can use some of those elements with the field portable, such as lead, zinc, manganese, possibly copper, but definitely now nickel as well. So the only one that's probably not that effective is maybe copper, but we're still working on that one. The laboratory results these days are fantastic. There's very low detection limits, so that's assisting with the importance of especially lead, tungsten and rhenium for vectoring towards mineralisation, and gold as well. If you're planning some biogeochemistry sampling, I would recommend that at the prospect mine scale level, you probably don't want to go any closer than about 25 metres up to about 100. I generally average around 50. That seems to be working pretty well. And then you can space that out to 200 to 400 for more wide space targeting to make sure you can find enough pines. Sometimes you get lots of pines in an area. Sometimes they're spaced out amongst the eucalyptus and other species. So today, with the support of MINEX CRC and their funding scholarship, New South Wales State Government via the Geological Survey of New South Wales, and the support of the explorers and miners in Cobar, we've spent nearly $200,000 to date in laboratory costs alone. So that's provided a fantastic database for us and gives us confidence in the numbers and the averages of the values of the elements in the trees that we're looking at. So these are some of the sponsors that have been pretty key supporters of the PhD work so far, and in particular, both Aurelia and Peel Mining. So thanks to Adam and Kidden at Aurelia and Michael Oates and the team at Peel Mining. Also Quintana Resources at Mineral Hill, Sunrise Energy Metals at the nickel cobalt scandium deposit at Firefield, 3E Steel with the Benbella Gold Project, Golden Cross with their Burra prospects south of Cambeligo, and Sky Metals, where we looked at the response of tungsten and tin in the needles at the Tallabong deposit. Lab West have done all our analytical work for our biogeochem samples, and Intertech Laboratories has done our check and QAC sampling work as well. And finally, thanks to Olympus, we've been working with them for the last 2 or 3 years, and for loaning the Vanta to do a lot of the test field work on the cypress pines. In terms of future plans, finalise and complete the database. We've got about 3,500 samples now for the pines, plus QA, QC, some soils. So it's probably about a 4,500 sample database. A lot of interpretation still to come, looking at the results, pulling all the database apart and looking at things like, you know, the response over faults, the response of pines to different underlying geology, like the granites and in the cover zones. We've also got an honours student up at James Cook University doing some pine isotope work for copper, lead and zinc at the at Aurelia's Federation deposit. We're working on the ongoing use of the field portable out in the field. So who knows, in the future we might be cutting the cones up and zapping them as well, as long as the needles and maybe some of the twigs for some of the elements. And then finally, put all our data into the New South Wales Geological Survey's MinView online database system, where everybody will at some stage be able to access all my data and see if through machine learning that we can, maybe with all the other data in there, the soils, rocks, water, drilling, that might throw out a few more areas that coincide with some of the geochem anomalies as target zones, and then finalise the research over the next 12 to 18 months. So thanks for listening into our end of year 2021 presentations. If you've got any further questions, please feel free to call me at j.schifano or email me at j.schifano@unsw.edu.au. Thank you.